Okay.
You get a medal, Dr. Cole.
Okay, apologies for anyone listening in for what you've missed already, but most of it will probably be indicative from what was on the slide. But we were just at the point where we were discussing PPCM, which is an idiopathic systolic dysfunction that occurs during late pregnancy and in the first 6 to 12 months postpartum. And this particular disease has a highly variable severity. Some women recover fully and some women develop end-stage heart failure. And as I mentioned a few minutes ago, this affects 1 in 900 to 1 in 4,000 live births in the US, and it's becoming more prevalent due to advanced maternal age. As you'll see here on the graph here, actually rates of PPCM are significantly higher in Kentucky than the US average. So despite all of these findings, the specific molecular mechanisms that contribute to pregnancy-induced growth are not well understood. And it's important for us to understand these because failure of the heart to adapt during pregnancy could promote pathology such as PPCM, or it could diminish the ability of the maternal circulation to meet the demands of the growing fetus. So therefore, if we understand the underlying mechanisms that drive pregnancy-induced cardiovascular complications such as PPCM, it will allow us to effectively target these in future studies. So in my lab, we have established a model for pregnancy-induced cardiac growth. And for this, we've used 12-week-old FEB NJ mice. And we have 4 main time points of investigation. We have a non-pregnant diestrus time point, a mid-pregnant time point, which is day 8 of pregnancy, late pregnant time point, which is day 16 of pregnancy, and a 1-week post-birth time point. And for the purposes of the studies today, we've performed extensive gravimetric analyses, histological analyses, serial echocardiography. We've performed cardiac multiomics. We've examined the transcriptome, the proteome, and the metabolome. And we've performed stable isotope resolved metabolomics. And so starting with the gravimetric data, it's not surprising, but during pregnancy, we see an increase in body weight, which peaks at late pregnancy, which is the point of maximal fetal growth. We also see increases in both heart weight and heart weight to tibia length at all stages of pregnancy and post-birth. We also see that pregnancy induces cardiomyocyte hypertrophy. If we look at the myocyte cross-sectional area here, which was determined via wheat germ agglutinin staining, we see a significant increase in myocyte cross-sectional area post-birth. And in combination with the previous gravimetric data and this data, this is suggestive of more of an eccentric hypertrophy at late pregnancy and more of a concentric hypertrophy at post-birth. Similar to the documented phenotype of physiological hypertrophy, we see that in our hands, pregnancy is not associated with any changes in cardiac fibrosis because we performed serious red staining to examine collagen deposition, and we see no changes between our 4 groups here during pregnancy. We also see, characteristic of physiological hypertrophy, that there's no significant changes in the expression of genes associated with the induction of the fetal gene program. We see no changes in AMP, BMP, and beta-myosin. We next perform serial echocardiography to examine changes in cardiac function and chamber dimensions during pregnancy and post-birth. And we saw significant increases in chamber dimensions in that we see a significant increase in left end diastolic and end systolic volumes at post-birth. And this was without any significant changes in ejection fraction. Not surprisingly, we also observed significant increases in cardiac output throughout pregnancy and post-birth. And this was mediated primarily through increases in stroke volume rather than heart rate. So this led us to wonder, what are the molecular mechanisms of pregnancy-induced cardiac growth? So we know that during pregnancy, not only are there changes in systemic metabolism, but there's also significant changes in cardiac catabolism, namely through reductions in glucose catabolism and increases in both fatty acid and ketone body catabolism. And a recent study by Zoltan Arani's group examined this particular reduction in cardiac glucose catabolism at late pregnancy, where they saw that pyruvate dehydrogenase kinase 4, or PDK4, which is an inhibitor of pyruvate dehydrogenase, was significantly increased in expression at late pregnancy. This was in addition to increases in the phosphorylation of PDH at the same time point. And through a series of experiments in this study, they found that this increase in PDK4 was mediated through changes in the pregnancy hormone progesterone. But the most intriguing finding from this study was that if they inhibited PDK4 with dichloroacetate, DCA here, they inhibited cardiac growth. And this highlighted that a reduction in glucose oxidation was important for cardiac growth. So, taking our 4 time points of non-pregnant, mid-pregnant, late pregnant, and post-birth, we recently performed RNA-seq analyses to determine pregnancy-induced changes in transcript levels. This heat map here on the right shows significant changes in all groups during pregnancy. In particular, we see that during late pregnancy, there are significant increases in metabolic transcripts, such as oh, my mouse has gone funny. Such as PDK4, which is increase in expression at late pregnancy, corroborating the previous Arani lab studies. And we also see distinct patterns in transcript expression between non-pregnant and mid-pregnant, mid-pregnant and late pregnant and post-birth time points. So taking our RNA-seq data, we performed supervised analysis using a partial least squares discriminant analysis here on the left-hand side, and this showed distinct group separation between all 4 groups. We next performed a variable importance in projection analysis, or VIP, and this shows the particular genes which precipitate this group separation here on the left. And we once again see, not surprisingly, that PDK4 is one of these genes that contributes to this group separation, and that it's increased at late pregnancy. And this is in addition to several other metabolic transcripts. In addition here, we see significant increases in other additional metabolic transcripts such as HMGCS2, SED1. We also see significant increases in several angiogenic transcripts at mid-pregnancy, such as MAP3K6 and angiopoietin-like 7. So if we interrogate both the non-pregnant and mid-pregnant time points and examine the top 50 differentially expressed transcripts, well, we are able to show that a significant upregulation in genes involved not only in cell cycle, cell migration, and cell motility, but also those involved in vascular development and angiogenesis. And here's just a small, several vascular endothelial cell markers that we had selected from the RNA-seq analysis that we see increased expression of at mid-pregnancy. We see significant increases in PCAM1, VEGF-A, VEGFR2, and VE-cadherin. And these are associated with the vascular compartment. And we also see here with the enrichment analysis several similar changes with respect to the vascular endothelial compartment, but also significant increases in those in cell cycle transcripts. If we move our attention briefly to the post-birth time point and look at the comparison between the non-pregnant and post-birth time points, we see of the top 50 differentially expressed genes, these show significant upregulation in genes associated with extracellular matrix remodeling and the presence of several different collagen isoforms. And this is just once again shown here in a little bit more detail, where we selected several of these collagen isoforms and periostin here, which show a small peak in expression at mid-pregnancy and a larger peak at post-birth time point. So turning our attentions back to the late pregnancy time point, of the top 50 differentially expressed transcripts between non-pregnant and late pregnant hearts, we see significant upregulation in metabolic transcripts. We see increases in PDK4 and oxyglutarate dehydrogenase, and also beta-hydroxybutyrate dehydrogenase 1, which appears to be increased throughout all stages of pregnancy and post-birth. And this seems to follow the arc of cardiac growth that we observe. So, of interest, as I mentioned previously, during pregnancy, there are increases in not only fatty acids and triglycerides, but ketone bodies are also increased. And they're at their greatest levels during late pregnancy. They're produced by the liver mitochondria and released into circulation and are metabolized by non-hepatic tissues such as the heart. And in addition to their roles as energy substrate, they have various non-metabolic signaling roles, but overall their role in pregnancy and pregnancy-induced remodeling is unknown. And several recent studies have highlighted the importance of ketone metabolism, cardiac growth, and metabolic remodeling. In particular, studies have shown that during heart failure, the heart can use ketone bodies as a fuel source to maintain function, and that the ketone body enzyme BDH1 may play a significant role in knockout and through the determination through knockout and overexpression studies. In addition, studies involving cardiac deletion of the mitochondrial pyruvate carrier showed that ketogenic diet could also reverse pathology. And collectively, these studies highlight the importance of ketone body metabolism in the heart. We also see in our data that there's a significant increase in blood ketone levels at late pregnancy. And this is also associated with a reduction in glucose levels at late pregnancy and significant changes in the glucose to ketone index, which is a measure of ketosis. And this changes in blood ketone levels and glucose levels are occurring at the time when cardiac growth is occurring during pregnancy. We also observe increased levels of cardiac BDH1 expression, and this, similar to the transcript levels, shows significant increases in BDH1 expression at all stages of pregnancy and post-birth. So future studies in the lab, which I had wished that I would be able to discuss today, but due to some colony issues, I can't. But we are in the process of generating mice with both cardiac-specific knockout of BDH1 and liver-specific knockout of BDH1 to look at the role of ketone body availability and ketone body oxidation during pregnancy-induced remodeling, but also potentially its role in the reversal of this remodeling. So hopefully if I speak to you next time, I'll be able to speak on these studies. Oh no. Okay. Sorry, it almost crashed then. So we next determined protein expression of key metabolic proteins that we identified at RNA-seq. We observed significant increases, once again, in PDK4 protein expression at late pregnancy, which is similar to the pattern that we observed in the transcript levels. We also observed a significant reduction in the phosphorylation and thereby activity of PFK2 at late pregnancy. And collectively, these are suggestive of a reduction in glucose catabolism. And this reduction in glucose catabolism at the level of both PFK and PDH could mediate coordination of ancillary biosynthetic pathway activity in order to save glucose-derived carbons for nucleotide and phospholipid synthesis, and it's also possible that ketone bodies could help maintain metabolism during this time. So to begin to investigate this, we performed stable isotope resolved metabolomics, in which mice were fed a liquid diet containing C13 labeled glucose for 18 hours, and this was following a acclimation period with a non-labeled liquid diet. And for the purposes of these experiments, we used our 4 groups, non-pregnant, mid-pregnant, late pregnant, but we also included an extra group here. And so we have a 1-week post-birth group with and without lactation. And to remove lactation, we did this through the removal of the litter upon birth. So we found specifically that pregnancy increased glucose-derived carbon allocation to nucleotide, glycerophospholipid, and amino acids. And this was despite no significant changes in the metabolite pools. And these data collectively suggest that increased demand for biosynthetic pathway activity end products is met in part by ancillary biosynthetic pathways of glucose metabolism. Interestingly, if you look at the graphs here, the highest enrichment was seen with the post-birth time points. And this increase in enrichment was significantly reduced with the removal of the litter in the post-birth non-lactation group. So these data suggest that the activity of several anabolic pathways that use glucose-derived precursors are augmented in the heart during pregnancy and are influenced by lactation. So we next went on to assess the cardiac metabolome, and we found that 492 significantly changed metabolites, and the majority of these belong to the lipid and amino acid superfamilies. We observed, if you look at the heat map here, of the top 75 changed metabolites, you'll see that there's relatively little difference between the metabolomes of the non-pregnant and mid-pregnant heart, but we see significantly changed metabolites in the late pregnant and post-birth groups. We see specifically for late pregnancy, this was associated with changes in metabolites associated with amino acid metabolism. We see changes in urea cycle metabolites, and including the urea cycle associated metabolite Homo Arginine, and several additional amino acid metabolism pathways. Of note, Homo Arginine was the most significantly changed metabolite of the whole metabolome, and this was increased during late pregnancy. We also see significant increases in fatty acid metabolites at late pregnancy, namely sphingomyelins and long-chain fatty acids. During the post-birth time point, we see a significant number of changes in the metabolome. And this is associated with similar changes to that we see during late pregnancy, in that we see significant changes in amino acid metabolism, namely that of the urea cycle metabolism and polyamine metabolism. We also see changes in nucleotide metabolism through purine and pyrimidine metabolites, and changes in glycerophospholipid metabolism through changes in phosphatidylcholines and ethanolamines. Supervised analysis showed, similar to the unsupervised analysis with the heat map, that there was significant overlap between the non-pregnant and mid-pregnant groups with the PLSDA, but we see significant separation of the late pregnant and post-birth groups from these. Not surprisingly, once again, we see increases in Homo Arginine driving these changes, in addition to several additional fatty acids and amino sugar metabolites. And also, we do see during the post-birth time point a lot of additional plant food components at this time point, and this is probably associated with the fact that at this time point the females seem to consume a lot more food. So, as I mentioned, the most changed metabolite in the pregnant heart is Homo Arginine, and this is shown here, and it's increased during late pregnancy. So what is Homo Arginine? It's a endogenous, non-proteogenic cationic amino acid, and it differs from L-arginine due to a methylene group. And similar to ketone bodies, it's formed in the liver in a reaction catalyzed by AGAP. And it's an alternative substrate for a nitric oxide synthase and increases the availability of NO and has been shown to affect endothelial function. So why is this also important to my studies? Well, not only do we see changes in Homo Arginine levels in the heart during pregnancy, it's also been shown that circulating levels of Homo Arginine are significantly increased during pregnancy for all trimesters of pregnancy in humans. And we also see here that the asymmetric dimethyl arginine, or ADMA, here which has opposing effects on Homo Arginine and is an inhibitor of Homo Arginine, seems to show reverse expression during pregnancy too. So utilizing our transcriptomic data, we observe significant changes in AGAT, which is the enzyme that generates Homo Arginine, along with several other key components of Homo Arginine synthesis and degradation. Also, utilizing our metabolomics data set, we observed several changes in key metabolites involved in Homo Arginine synthesis and degradation during pregnancy and post-birth. In addition, we observe, as I previously mentioned, several metabolites associated with the urea cycle that change during pregnancy and post-birth too. And so low levels of Homo Arginine have been associated with adverse cardiovascular events, such as cardiovascular diseases, and also it's been associated with preeclampsia. Also, Homo Arginine supplementation has been shown to improve blood glucose regulation in diet-induced obese mice. So future studies in the lab will be performed to analyze the importance of this increase in Homo Arginine in the pregnant heart, and some of these studies could utilize the cardiac-specific AGAT1 knockout mice that are currently available. So next time I visit, I'll also inform you on how those studies are getting along. So we next analyze the cardiac proteome during pregnancy and postpartum. And we see during non-pregnant and mid-pregnant, they also, similar to the metabolomics, show a significant overlap in some of the changes there. But we do see during late pregnancy, there are significant changes in proteins that have previously been shown to be increased during pregnancy, such as coagulation factor 5, ceruloplasmin, alpha-fetoprotein, and prolactin. But we also see changes in late pregnancy and post-birth in several metabolic proteins. Once again, we see PDK4 is significantly changed, SCP2, and several other metabolic proteins are changed in late pregnancy and post-birth. But the most significant finding of the cardiac proteome was that at late pregnancy and post-birth, we had significant reductions in the expression of several mitochondrial complex subunits. And these were mainly of cytochrome C oxidase and NADH ubiquinone oxidoreductase subunits. And also we see significant reduction in several histones and contractile proteins. So in the lab, we're also performing studies to interrogate the mitochondrial subunits and also mitochondrial function experiments to examine the impact of pregnancy and postpartum on mitochondrial function. And so when we perform supervised analysis once again of our cardiac proteomics, we see distinct group separation between all 4 groups. And we see several of the proteins I mentioned on the previous slide that contribute to the group separation here. Several changes in histones, laminin, B receptor, corticosteroid binding, globulin, and alpha fetoprotein, which I've shown to previously change. But one of those which is associated with the urea cycle in particular is dimethyl arginine dimethyl amino hydrolase 1. And this metabolizes ADMA, which inhibits nitric oxide synthase. So this also adds a little bit more weight to the Homo Arginine story that we have. So that's should be exciting. So when we performed full proteomic integrative pathway analysis, we see that several pathways associated with nucleotide, lipid, and amino acid metabolism are impacted in the heart during pregnancy and postpartum. In particular, we see significant pathway impact in the arginine and proline metabolism, which is part of the urea cycle, which is associated with Homo Arginine. And collectively, these data are supportive of increases in metabolic pathways associated with cardiac growth. So far, a brief summary here is that during mid-pregnancy, the heart enlarges, and this is primarily through, we see primarily changes in angiogenesis, cell cycle, and cell proliferation, and increases in BDH1 expression. We see a further increase in heart size at late pregnancy, which is associated with increases in, changes in metabolism, in that we see increases in PDK4 expression, decreases in phosphorylation of PFK2, and sustained BDH1 expression. By post-birth, the heart is still enlarged, but we see changes in extracellular matrix markers and reversal of some of the metabolic changes observed during late pregnancy, in that we see a reduction in PDK4, but also a sustained BDH1 expression. And between these 2 time points, we also see increase in pro-growth metabolites in amino acids and in the form of those urea cycle associated metabolites, glycerophospholipids, nucleotides, and also ketone bodies. Having defined the growth processes occurring during pregnancy, we next asked, when do the pregnancy-induced cardiac adaptations and hypertrophy reverse following birth? So it has been shown in several studies that heart size appears, these studies are in mice, sorry. So in these studies, they've shown that within 7 days of birth, that the heart weight, body weight, and heart weight to body weight ratios appear to return to non-pregnant levels. However, many of these studies, it's not clear whether these dams were allowed to lactate or not. Also, in a rat model of pregnancy adaptations, they also observed within 2 weeks post-birth that heart size had returned to non-pregnant levels. So this made us wonder, what are the processes occurring in the postpartum heart that give rise to a reversal of pregnancy-induced growth? So following on from our earliest stable isotope resolved metabolomic study, in those studies we found that heart weight was significantly increased at post-birth and significantly reduced with post-birth non-lactation. But due to the sample collection, we couldn't collect hearts under that circumstance. So we performed an additional study to gain gravimetric data and histological data. And we found that similar to that observation, we see an increase in heart rate throughout pregnancy and post-birth, but this heart rate is reduced upon removal of the litter to similar levels that seen during late pregnancy. And this is associated, although it's not a significant change here, it's associated with a trend for a reduction in myocyte size. To also avoid any potential issues with mouse strain, we performed this experiment with C57 black 6 mice as well. We see a trend for this increase in late pregnancy and heart rate to tibia length, but we also we see similar changes, but we have to investigate these further because these studies were not ideally powered. So off the back of the study where we saw a reduction in heart size in the post-birth non-lactation group, this led us to design the following study. We have our non-pregnant, and then we have various time points during post-birth. 1 week post-birth, 3 weeks post-birth, then the litters are weaned at 3 weeks, and then we followed the mouse out to 6 weeks. But we also did a parallel group where we removed the litters upon birth, and then we followed these out for 1 week, 3 weeks, and 6 weeks. And so, just before I get into this, I'd like to point out that the dotted line represents the non-pregnant average on the next few slides. So we see the postpartum females remain significantly larger following birth, and this is irrespective of lactation status. And we see, although we see no significant changes here in any of the circulating metabolites. We do see that heart rate remains elevated up to 6 weeks postpartum. And we see here on the left-hand side, biventricular weight is significantly increased 1 week and 3 weeks post-birth. And this difference is non-significant by 6 weeks. However, I'll draw your attention to the average of the non-pregnant. Although the influence of lactation here has been removed, the heart size has not returned yet to non-pregnant values. And this is also the same with the atrial weights and the total heart size. So additional studies that were ongoing in the lab at present are to examine the point at which the heart returns to non-pregnant size. And to do this, we'll be using serial echocardiography to follow cardiac remodeling out and to be able to gain some hemodynamic measurements there too. And so we also see similar changes in the expression of the heart in the post-birth to those occurring in the liver. And this, we see a significant increase in liver size post-birth for 1 week and 3 weeks. And by 6 weeks, there's no significant change between the post-birth and post-birth non-lactation groups. And this is significantly increased versus the non-pregnant groups. And we see some additional changes in other organs during pregnancy, but for the most part, most of the major changes seem to be occurring at the level of the heart and the liver. So this is taking us back to this previous summary diagram. If we go to 6 weeks post-birth, we know that the heart is still the same size as that maybe of mid-pregnant, and it hasn't fully returned to non-pregnant size. And this decrease in size from post-birth is due in part to changes in lactogenic status, but also we think that it could be due to changes in both PDK4 and BDH1 signaling there, but also it could be changes in some proteins known to participate in atrophy signaling. And so hopefully I've shown that pregnancy-induced cardiac growth and its reversal involve coordination of several distinct signaling processes, and changes in metabolism play a significant role in pregnancy-induced cardiac growth. And so there are several questions that remain. And so if we bring our attention back to the RNA-seq findings, where we see at mid-pregnancy there's more of a change in the vascular compartment, late pregnancy there's more of a change in cardiomyocyte metabolism, and post-birth there's more of a change in extracellular matrix remodeling. This suggests that there's an extent to which other cell compartments play a role in pregnancy-induced remodeling. And also, given the fact that many of the major metabolites that are highest in circulation that seem to contribute are derived from the liver, namely ketone bodies and Homo Arginine, we need to determine the role that they play in pregnancy-induced remodeling. We also don't know the full impact of the circulating hormones, the placenta, and any maternal-fetal interactions that could impact this remodeling. And we also don't know the extent to which ketone bodies and some of these additional changes may play in remodeling and how they could play a role in PPCM pathology. So with that, I'd like to acknowledge and thank my faculty members and collaborators and research course and personnel in the Diabetes and Obesity Center and the funding sources that have made this work possible. And I'd like to take any questions that you may have, and I thank you, and I apologize for the technical issues. And here is today's event code.
That was fantastic. I mean, you've done so much work and you have so many different components to come together with your multi-omics approach at the different animal numbers. I was intrigued by one finding in your RNA-seq data, and that was UCP1. But I wasn't, there was so much going on. Was that UCP1 upregulated in the post-birth, or was that in the late pregnancy?
Oh, goodness.
It was in the heart, so...
Yeah, I think it was late pregnancy, but I have to check back. There was so much going on that diagram. There's also changes in UCP3 too.
Interesting.
So
I think about that in brown animals. I find something skeptical. So are there brown cells in the heart? I mean, you took heart and did that, right?
Mm-hmm. So. Yeah. We took heart and did that. So we need to, one of the planned experiments that we were going to do is do a targeted approach and do some protein expression analyses, because as most people are aware, not all transcriptomic changes correlate to protein changes. Right. So we have plans to look at UCP1 and UCP3 and several of the other metabolic markers there. Now I think there might be an issue for some of those finding a good antibody, but yeah. And we can also actually go back in the proteomics and see if that's changed there. Yeah. That will probably be faster actually to see.
A lot of these changes.
Mm-hmm.
How long?
So our post-birth time point is 1 week post-birth. And so most of the data that I showed, they're 1 week post-birth and they're allowed to lactate and have their pups with them. And so part of the last part of the talk is to try to investigate when that increase in cardiac size reverses to non-pregnant levels. So we had seen that within 6 weeks, the effect that lactation is having on cardiac size is removed. So we don't see any difference between post-birth and post-birth non-lactation. But we do see that post-birth and post-birth non-lactation hearts are significantly larger than that of non-pregnant. So we are currently in the process of performing some studies where we take mice out for longer than 6 weeks via the use of echocardiography to see if they do reverse back to normal levels. It's possible that they don't, but we won't know until we've looked at that in more detail. But in the human, it's thought that following lactation and within about a year, cardiac hemodynamic parameters, echocardiography parameters seem to be that of non-pregnant levels. So we need to interrogate that further in future studies. Yeah. So lactation seems to be, I guess, here preventing the reversal of hypertrophy. Yeah. So that would be beneficial because there's going to be still a increased demand on the mother during the lactation period. So, we just stumbled upon doing that. We were doing the stable isotope resolved metabolomics, and I thought, well, because this experiment is so expensive, I need to make sure I've got all the groups that I need. And I was like, well, yeah, I need to remove the litter to see the effect there. So we need to interrogate that in more detail. We've started to perform some XF studies looking at mitochondrial function, but in the presence and absence of certain hormone combinations. At the moment, we're actually seeing data that's suggestive of a reduction in mitochondrial function with some of these hormones. So we need to look at that in more detail.
It corresponds to some of your metabolic data as well.
Yeah.
Away from the T.
Check if there is.
Good question.
Oh. Okay. What did you say? Sorry.
So delayed pregnancy, permanent God visa mice. Human pregnancy, placental age, like the glucose. I didn't know if.
So I guess putting the differences in gestational length aside, I guess our late pregnancy time point would be most reflective of that of the final trimester of pregnancy. So obviously there's going to be some species dependent differences there, but that would be indicative there of that time point. Does it say? Is there any particular stage during pregnancy that the chances of death are higher, and what could that be due to? Okay, so there's a number of studies that have shown that if female mortality is higher during late pregnancy and post-birth. And there's been some studies that have shown that some of these ladies can develop some arrhythmias, and also some of the pregnancy-induced cardiovascular complications. So some women develop preeclampsia, some women develop PPCM, but there's not really been a complete study that has looked at all of the death. what's the word I'm looking for? Reasons for death in these populations that I know about. But
One of the hypotheses, mortality, age. So in the mindset, they are older, reversible changes or
so I did start out to try and do an aging study. We probably need to go back and obviously do that again, but the issue I was having in the mouse models in comparison to the human models is that they seemed, maybe it's our mice, but by about 8 months of age, I was not really getting many litters or anything. So it was tricky at that time point. What I want to do is, because most of the animal models of PPCM, and this is also reflective of human studies as well, it usually takes more than one pregnancy to induce PPCM as well. So it's usually on the second or third pregnancy. So I wanted to do a study where I look at the effect of number of pregnancies on that, because I think if our data show that the heart doesn't go back to normal size following pregnancy, and it's always got a higher baseline each time, that could be one of the reasons why. But also, I don't think I mentioned it, but PPCM is also associated with a lot of angiogenic changes, namely anti-angiogenic. So that's also something that we need to consider moving forward as well, because a lot of those changes in angiogenesis can be impacted through age too. I don't see any more questions online. I think I made good for the time.
You did. Well, thank you very much. It was really a great talk.
Thank you. Hopefully I can update you again next time.
Okay, I'll put you on my list for invitation next year.
I don't know how to, I guess I have to end call.